Okay. Hey, everybody. So for this video, I want to go over some extra details that weren't explicitly mentioned in uh, the other videos that you've seen. And this is mostly to prepare you for the cumulative uh, lecture final. So yeah, it's just a few additional details here. Nothing too different or new. It's all extremely relevant to what you've already learned. It's just whenever you have a standardized exam, there's, one person makes it, and of course people contribute and whatnot, but it's hard to have a universal way to assess everyone because everyone teaches differently and maybe mentions a one word that others don't or a term. So I just want to make sure we're all fully prepared. And so yeah, let's jump right into it. Here we go. Okay. Extra details. Now, first up, I want to talk about mutations. Oops, sorry, hit a button. Okay, I want to talk about mutations a little bit. Just we've talked about mutations already. We know what they are. They are simply a change in the DNA sequence, those nucleotides, the order and arrangement of nucleotides in a DNA sequence in a DNA molecule on one of the strands. So any change is a mutation, but I want you to be aware of the different names for different mutations that can occur. So the first is actually a category. There's really two categories of mutations. As far as these what we can say small micro mutations where there's like a single nucleotide change or maybe one or two nucleotides. It's not like a large chromosomal crazy really harmful mutation in that regard, although these can still certainly be really harmful to an organism. So this first category is called point mutations. So a point mutation. All right. So point mutations. And so a point mutation, I'm actually gonna just, my bad, let's just say point mutation. So a point mutation is a change in a single nucleotide of a DNA sequence. Hence why it's a point. So it's just one nucleotide that's going to change. And by that, I mean, really, it's the base. So it's like an A to a C or an A to a T or a T to a G, whatever it is, it's going to change. And so really, the different types of point mutations, it all has to do with the result. It all has to do with the result, meaning the result of what that single nucleotide change is going to, or how it's going to affect the final protein product. Meaning, the sequence of amino acids, and really more specifically, the codon. How is it going to change that codon that at the ribosome is going to determine what amino acid is brought in when assembling the protein? Okay. So let's start with our first type here, which is a silent mutation. Okay, so let's start with a silent mutation. Okay, silent mutation. Yeah, what's this? All right, so if you have a silent mutation, which is a type of point mutation, like the name implies, it does not affect the amino acid sequence, does not affect the amino acid sequence of a protein. Really meaning that the codon is going to change, of course, because you're changing one of the nucleotides. But this has to do with redundancy in the genetic code. In fact, let me bring up a genetic code image. Sorry, this is taking a while. Okay. So what we're talking about here is when a single nucleotide change in DNA, remember mutations are in the DNA, well, that could result or will result in a different codon in the mRNA. So codons are mRNA that are transcribed from DNA. So let's say, you have CCC and then it changes to CCA. We would consider that a silent mutation because both of these codons still code for proline, this amino acid proline. So when translation is occurring at the ribosome, it's almost like it goes undetected. It's silent because the same amino acid is transferred in, and you get the same protein at the end of the day because of that. So that's what we call a silent mutation. But of course, this isn't always the case. Sometimes the amino acid change, and it shows this here as well. But the next one that we can, or that can happen is called a missense mutation. So a missense mutation it's definitely not silent. It results in a different amino acid being incorporated into the protein. Into the protein. So it's a change. the original amino acid that's supposed to be transferred in during translation at the ribosome. It's the codon has changed so that it actually codes for a different amino acid. So the slide, take a look here, shows an example of this. So in this case, you can see that, let's see, here we go. See this highlighted A. So this CCC codon that coded for proline, well, now it changed, the C changed to an A. You can see that change there. Well, ACC, Arapaho, ACC codes for threonine, an entirely different amino acid, not proline. So we would call this a missense mutation. Okay. All right. Last up, we've got a nonsense mutation, which it screws it up so much, it just doesn't make any sense anymore. So a nonsense is our last type of point mutation here. And a nonsense mutation converts a codon into a stop codon. Let's see that. Into a stop codon. And of course, a stop codon stops translation altogether, signals to the ribosome a protein called a release factor is brought in to stop translation altogether. And so, of course, this would leading to premature termination of protein synthesis. I don't know if I can fit that here, but to premature, yeah, definitely cannot. So let me erase a bit. Premature termination of protein synthesis, which is never good. Sometimes, like a missense mutation doesn't affect the protein structure too much. It can, especially if it's in an important part of the protein, like the active site of an enzyme, for example. It could definitely, remember that lock and key kind of enzyme substrate specificity. It's got to be exact. But if the missense mutation codes for an amino acid with similar properties and it's on the outside of the protein or something, it might not affect it too much. This is always going to be really bad, almost always, because if you got an amino or a protein that's 400 amino acids long, you know, some kind of structural protein or an antibody defensive immune system protein, whatever it is. But then the stop codon happens at like the nonsense mutation happens at like amino acid 200. Well, you've still got 200 amino acids left and it's stopping. So you get half your protein, which almost always isn't going to function properly and do the proper function that's needed by the cell. Okay. Any question? Well, I was going to ask for questions, but this is not live, so just kidding. But email me, message me, whatever. Okay, so there's one more category of mutations I want to talk about, which is our insertion/deletion mutations. So this isn't a change in a nucleotide. It's adding an extra one, usually one or two. But it's adding an extra one, or it's deleting one, getting rid of one. In this example shown here, there's an insertion. So you see the ATG, CAT, TGC. Well, now it's ATTGCA. So it inserts a nucleotide here in between this G and C. And the classic catastrophic example of an insertion or deletion is called a frame shift. Okay. So, well, first let's define insertion, deletion, mutations. Insertion/deletion mutations. So this involves the addition or removal of one or more nucleotides in a DNA sequence. Okay, and the example of an insertion, deletion, or indel for short, that I want to talk about is called a frame shift mutation. So a frame shift mutation. Okay, so let me explain what a frame shift is first, and then we'll write it in. But you might have noticed here that the insertion of this extra T in between the G and the C, it caused a frame shift mutation. And the reason is whenever the insertion or deletion isn't a multiple of 3, it shifts the reading frame. And by that, I mean, remember that every 3 nucleotides of DNA gets converted, or not converted, transcribed to produce messenger RNA during transcription. And, you know, you'd have UAC and GUA and whatever. Well, that's the normal sequence. But if you do this, you have changed the codons downstream. The remainder of your codons is completely shifted. So now this is ATT, that's different, but then it's no longer going to be CAT. The triplet groupings have changed. You've shifted that reading frame, which of course is never good because all the remaining codons are going to code for different amino acids, most likely, right? So frame shift mutations are no bueno. And so to define this, we can say if the number of inserted or deleted nucleotides is not a multiple of 3. It causes a shift in the reading frame. Which arrow or comma or whatever, altering, so it's going to change all downstream codons. So it almost always produces a non-functional protein here. All right, that's it for mutations. Next up, I want to talk about, really briefly, just a couple, again, extra details about related to gene regulation, related to prokaryote versus eukaryote transcription and central dogma and kind of the differences. But really, it's just this this fact that in eukaryotes like us, after messenger RNA is produced by transcription, it undergoes this splicing process. So it's mRNA processing, whereby, weirdly enough, I was reading about it last night just to familiarize myself with why this happens and kind of, but there's really, it's still definitely an ongoing area of investigation for sure, but we have some idea of why this happens. And essentially what's happening is when you have a gene, which is DNA, there's actually, and remember, genes code for proteins, as we know. Well, there's actually stretches within the gene, sequences within it, that aren't going to be part of that final protein product. We call those introns. So you can see after the messenger RNA is produced, you have little stretches of mRNA from the DNA called introns, shown here. But you also have stretches called exons. So you've got introns and exons. Before the messenger RNA goes to the ribosome to be translated, the introns are removed, which is annoying because you'd think introns stay in. Nope. Of course, it's the opposite. The introns don't stay in. They get spliced out. They get removed from the messenger RNA molecule. And then the exons get combined and put together. So that mature final messenger RNA is only the exon regions together. So yeah, I was reading a bit about this, and again, a lot of interesting complexity here. But the one I was reading, the information I was looking at mentioned that because there's these non-coding regions of introns, remember we learned about crossing over, which is kind of fun. You get to connect that topic here. If crossing over happens in a region that is an intron, you can actually do what we call exon shuffling, which the exons of one allele can get put with the exons of another and kind of create new protein variants. It's just another way to generate diversity and possible new protein. Anyways, that was just one evolutionary significance that I was reading about. But just knowing this is sufficient. Okay, next up, another quick fun fact. Again, none of this is like brand new foreign craziness. It's just kind of an extra little detail. And it has to do with cytokinesis between plant cells and animal cells. And there's just this subtle difference because, as we've learned, plant cells have this external structure surrounding the cell membrane called a cell wall made of cellulose, while animal cells do not. And so when the plant cell divides, we'll start there, it has to form a new cell wall to connect the two brand new cells. And so that structure is called a cell plate. So a cell plate is formed in order to split the two cells during cytokinesis. Whereas in the animal cell, we do a slightly different way of splitting the cell. During cytokinesis, we actually form a ring of contractile protein fibers, actin and myosin. It's the same for our muscle, actually, that contract. And we contract and we squeeze the middle of the cell, which causes this shallow groove that we call a cleavage furrow. And then eventually it squeezes the cell or contracts around it enough that it pinches it into two. So it forms that cleavage furrow, and you have the contractile actin ring versus a cell plate. Just a subtle difference there. Okay. Next up, related to organelles and structures within the cell are what we call cell-cell junctions, meaning structures that are between different cells that help them adhere to one another, or even transfer molecules to one another directly by that contact. And again, it's going to depend on the function of the cell and what it's doing. There's different types of junctions. I want to focus on the ones in animal cells. There's really three different main types of cell-cell junctions in animal cells. We'll start with tight junctions. So tight junctions. So as the name implies, these are junctions that keep cells very tightly bound to one another. And it's specific proteins that help, that are involved or make up that tight junction. And the key thing that they're doing, kind of structure and function connection here, because they're so tight, they're creating a seal. They're creating a barrier. So they establish a barrier. Establish a barrier that prevents leakage of extracellular fluid, so fluids surrounding cells, and actually we'll just keep it there. So classic example is our skin cells. Skin cells are all connected by tight junctions to prevent water loss from the connective tissue, underlying connective tissue. which is good. We don't want to lose too much water. So that's one example. The next we have desmosomes, and here's a cool image showing this. Here you have the tight junctions preventing fluid loss. Desmosomes are next. Desmosomes are a little interesting. They have a similar kind of function, but slight difference. These are like anchors to me. The desmosomes are like anchors. So you're gonna find desmosomes in cells that endure a lot of stress and force and tension. So the desmosomes attach cells that endure high tension and stress. And we'll there. Hi, hey. Sorry. Okay, I think she's done. Yeah, people outside are making noise, so I think she got bothered by it. But anyways, the desmosomes. Yeah, so you know, the classic example are muscle cells are constantly gonna be generating or under stress and tension. And to me, so the desmosomes kind of function like shock absorbers in a way. And the desmosomes, you know, they're on the surface of the cell at the plasma membrane, but they actually connect to cytoskeletal proteins, cytoskeleton, to kind of help act as like a shock spring or shock absorber if the cell is getting forced on it. If I have a weight or whatever, and that force is pushing against my muscle cells, and it kind of can act like a spring in a way. That's how I think of them. The last one we have has a very different function, actually. The gap junctions. Gap, as the name implies, gap junctions are going to allow cells to communicate to one another via molecular transport and move different molecules between one another or ions. So our classic example, as we'll see, is heart muscle, cardiac muscle has gap junctions connecting the different cells, the cardiac muscle cells. So the gap junctions provide cytoplasmic channels cytoplasmic channels from one cell to an adjacent cell, allowing for continuous movement of ions. It could be sugars, amino acids, so nutrients, etc. Yeah, and if you take anatomy and physiology, you learn that when it comes to the nervous system and action potentials, stimulating muscle contraction, and the rhythmic contraction of the cardiac muscle in particular, they do a special kind of way of transporting ions from one cell to the next, kind of this continuous flow. It's they have that connection between one another. It's like a tunnel, you could say, between two cells, to help this rhythmic kind of synchronized contraction. And they have those gap junctions. Okay, what do we got next here? Okay. So hierarchy of classification. Yeah, so this is kind of just really the hierarchy of classification is how we group organisms, plain and simple. And of course, we don't get into this too much in this class because it's 1111 is a cell molecular focused class. So the fact that this is on the exam is kind of interesting. But it is how we group organisms. And, to be fair, we look at microscopic organisms that are in different orders and classes, and we definitely name them with genus followed by species. So, yeah, essentially, the way this goes, I like this image because it shows that the domain is the most broad, the most all-encompassing when it comes to classifying organisms. And then as you go down, you're getting more specific when it comes to your group. So we have 3 domains of life, only 3, but oh my, there are so many different organisms and classes and phyla and all these things. But in this red fox example, the fox, it's in the domain Eukarya, it's eukaryotic. It's an animal, so it's in kingdom Animalia, which is within this domain. It's a chordate, so it's in phylum Chordata. It's a mammal, so it's not a reptile. It's not in Reptilia, but it's in the class Mammalia. Carnivorous, so it's order Carnivora. Family Canidae, genus Vulpes, species also Vulpes. So look at that. So yeah, that's really the key here. And to help remember this, because on the exam, a question could be like, which of the following is the most specific, or which of the following includes the others, includes all the others, like that are listed there, meaning which one is the most broad that are listed. So a good way to remember this okay, so dear King Philip cried out for great soup. He really wanted that soup, so he cried out for it. So domain, kingdom, phylum, class, order, family, genus, species. And on top of, when I teach this, I also like to mention the most specific classification is the species. And really you can identify an organism kind of like a first name and last name by its genus and followed by its species. Humans, us, we are in the genus Homo. And when we write the genus and species, for some reason, we're very particular about how we format it. So we always italicize or underline. So Homo, our species, sapien. And then, so sorry, the genus is capitalized, and then the species is lowercase. So Homo, also italicized, sapien. Which can also, you can also abbreviate the genus, H, sapien. So we call this the scientific name of an organism. And every organism has a scientific name. Like my barking dog is technically a wolf. She acts like one, though. But Canis lupus, bacteria, if you take microbiology, which a lot of you are going into nursing and medicine, and you'll be taking that, you'll learn all about, oh my gosh, tons and tons of different, Staphylococcus aureus, genus and species of a lot of different bacteria. Streptococcus pneumoniae, Neisseria meningitidis, there's a bunch of different ones, different species. And the last thing I like to say about this is, there's typically a common name for organisms, like human, for example. But if I'm communicating with a scientist or working with a scientist in Russia or Argentina or whatever, if I say human, they're not going to know what I'm talking about. They have a different word for that. If I say monkey, if I say dog, they have a different word. But if you say Homo sapien or Staphylococcus aureus, they'll know exactly. So the scientific name is universal. It's a universal language of identifying organisms. Okay, moving on. Just a couple more things here. All right, so we did talk about the different examples of theories, but I just want to expand on them and just tell you what they are, or rather, what they say. And the first is our cell theory. Cell theory has 3 tenets to it, which are life. All life is made of the fundamental unit. It's these little cells we've been talking about all semester. Second, cells form the basic unit of structure and function in organisms. As we know, even today, we've just been looking at how cells provide tight barriers and tight junctions for skin or muscle contraction, whatever it is. They're going to be that essential unit that directly provides the mechanism of structure or function for organisms, and of course defines the function structure. And then, of course, the last one is all cells come from pre-existing cells. All cells come from pre-existing cells. That would be our third. A lot of data to support all of this, as it is a theory. The second one that I mentioned at the beginning of the semester is called germ theory. Essentially, plain and simple, the germ theory, a lot of scientists first I showed this hundreds of years ago, actually, like Louis Pasteur, which is that microorganisms, like those bacteria I was just talking about, cause infectious diseases. They are the reason. So definitely a lot of evidence to support that. And then our last theory here I want to explain is gene theory, which really, when we're talking about mutations, that's gene theory. It is the genes are determining the proteins, technically, or characteristics of living organisms. And then, of course, this genetic material is what's passed on. The DNA is passed on. And yeah, if you get a mutation, if it's actually one of the cells that gives rise to gametes, of course, that's going to be passed on to offspring. And that is all detailed in gene theory. Okay, almost done. So next up, isomers, just a basic definition here. We talked about glucose, galactose, fructose as these carbohydrates. But as you can see here, they have different structures, molecular structures, but they have the same chemical formula. Each one. Each one has the same. So what do I mean by that? C6H12O6. They have the same number of atoms and same number of different types of atoms, more specifically. But as you can see, they have different structures. And so that's the key with isomers. They have the same chemical formula. Let's actually write that down. I don't like to be too PowerPoint-y, as you know. So isomers have the same chemical or molecular formula, but are different in structure. Another term that I might have mentioned, can't remember though, so I want to make sure. Amphipathic. So amphipathic molecules, they're kind of dual natured. They have a hydrophilic portion and a hydrophilic and hydrophobic portion. So it's one molecule, but they have different affinities for water, essentially. Or you could even say charge or polarity. They've got now I hesitate to say polarity because water is polar. It has opposite partial charges in different parts of the molecule. But really what we're talking about here is difference in, you have a charged area and a neutral area. Classic example of this was our phospholipid. Remember, our phospholipid had, oh my gosh, thank goodness, another color I can use. Remember, our phospholipid had that hydrophilic phosphate head because it had a negative charge. But the tails, the fatty acid tails, had no charge, and so they actually repel water. Water would rather bond, hydrogen bond to itself, has no attractive affinity for the hydrophobic fatty acid tails. Oh boy, it's gonna work. Hydrophobic. Oh, okay. Last thing. And I forget as well if I mentioned this in when we went over cellular respiration. But I do sometimes, and it is a cool, interesting point. But, and I can draw it out again here. It has to do with the purpose of fermentation, like why cells ferment. And if you remember that flow chart that we do, every cell, no matter what, starts by converting glucose to 2 pyruvate molecules. This process, this pathway is called glycolysis. Okay. And remember that from glycolysis, and actually I'm going to emphasize something else here really quick. So remember that during glycolysis, you get, or by the end of it, sorry, I'm scrambling my words. By the end of glycolysis, you get the 2 pyruvates. You also get 2 NADH molecules and 2 ATP, 2, 2, and 2. So you get 2 NADH, and remember, to get that NADH, you need, or rather 2 NAD pluses come in as coenzymes and help the enzyme in that step, happens twice, oxidize the glucose, that substrate at that point, which is a glyceraldehyde 3-phosphate. It's that 6th enzymatic reaction, which happens twice, but you don't have to worry about that. The point is that 2 come in and are converted. They take on 2 electrons and a hydrogen ion each to form 2 NADHs. Okay. You also get, on this green, sorry. You also get 2 ATP. So 2 NADHs, 2 pyruvate, 2 ATP. Okay. Then, as we learned, the pyruvate can be directed to the aerobic or the anaerobic pathway. Right now, I want to focus on the anaerobic pathway. And I know it's been a while, so I want to wrap this up soon. Essentially, the pyruvates, when you go the anaerobic pathway, which means no oxygen, oxygen is absent, that's fermentation. Fermentation, of which there are 2 main kinds, alcohol, lactic acid, depending on the organism. But what I want to focus on is why. Why do this at all? What's the point? In biology, there tends to always be a reason why. It doesn't just happen for no reason. Well, look, we do glycolysis, these organisms, these bacteria that ferment, or our muscle cells under intense exercise, we're going to ferment. But the reason mainly, right, we want this. This is why we do all this. The whole reason we even lecture on this is because it's how cells generate ATP molecules, energy. Why do this? We got our energy already. We did our glycolysis, we got the ATP. Why are we continuing anything? The reason is because during fermentation, when pyruvate is actually converted to either lactic acid or ethanol, the following happens. The two NADHs from glycolysis are used, and they reduce the pyruvate, meaning electrons and a hydrogen ion are transferred to pyruvate twice, because there's two, but point is, you're going to convert this NADH back, to two NAD pluses. Why do you think that's so important? You got it. Whoever's listening still, you need those in order to do glycolysis again. If you only have NADHs, you cannot do this process again. You are stuck with only two ATPs. But guess what? You want to keep doing this over and over and over. You want to get 2 and 4 and 6 and 8. You want to keep producing ATP molecules. Yeah, 2 is not enough. You got to keep doing this. And the only way you can do glycolysis is if you have NAD+. And yeah, so I tend to call this regenerating redox potential. Redox potential, your potential to undergo a reduction oxidation reaction and oxidize glucose ultimately and form these NADHs. I also sometimes say you're reloading the glycolysis gun. Like if you don't do this and you just have NADHs around, you can't fire off glycolysis again. So that's the primary purpose of fermentation, is to restore your redox potential and your, in other words, your NAD plus molecules. Woo! That's all I got. Okay. Yeah, that's it. So hope that helped. Let me know if you have any questions, and yeah, good luck.